Hi, my name is Jaspreet and today I will be talking about the most common type of iron deficiency known as iron deficiency anemia. Let's look at the word anemia and break it down. The prefix "an" refers to lack and "emia" refers to blood condition. Anemia essentially means lack of blood, so medically it refers to a low hemoglobin count or low hematocrit. So there are particular numbers for the cutoff for the hemoglobin or hematocrit to make a diagnosis of anemia. Now, these numbers are going to be different in different references, but for the most part, we are going to see that with hemoglobin, if it's less than 130 grams per liter in males, that would be considered anemia. And if it's less than 120 grams per liter in females, it will be considered anemia. And with regards to hematocrit, which is the ratio of red blood cells in blood, if it's less than 0.41 in males and less than 0.38 in females, it will be considered anemia. Red blood cells are also known as erythrocytes. Now, what's important about these red blood cells is that they contain a protein called hemoglobin, which is important for carrying oxygen to the other cells and tissues. They can also carry carbon dioxide from the tissue back to the lungs. It has an average lifespan of 120 days, and iron is required for producing the red blood cells. So if there is not enough iron, we are going to get an iron deficiency anemia. Now, let's move on to the essential proteins. Ferritin is a key protein that plays a central role in iron metabolism. When iron is absorbed from food, it is stored inside ferritin molecules. If iron levels begin to drop, the body uses up the ferritin stores first, which is why low ferritin level is often the earliest sign of iron deficiency. Another important protein is transferrin. It is a blood plasma protein that binds to iron into the bloodstream and transports it to where it's needed. In case of iron deficiency, transferrin levels usually go up because the body is trying to capture and transport as much iron as possible. So why is anemia important? This condition is particularly important in both pathology and clinical medicine as it affects a large portion of the population, especially women and children. In iron deficiency anemia, the body lacks the important iron needed to produce this important protein, leading to reduced oxygen delivery to the body's cell. And that's when symptoms like fatigue, weakness, dizziness, and shortness of breath can appear. When the body doesn't have enough iron to produce hemoglobin, it directly affects the structure and appearance of the red blood cells. Another very important finding in iron deficiency anemia is when looking at the complete blood count. It's looking at the mean corpuscular volume or MCV, which is the average volume or size of the red blood cells. MCV less than 80 will be found in iron deficiency anemia, which indicates both microcytic and hypochromic red blood cells. So, this means when we look at the blood smear under a microscope, it shows red blood cells that are smaller than normal size and the red blood cells appearing paler than normal because they contain less hemoglobin. To diagnose iron deficiency, several important pathology tests are used. The first and most common is the full blood count. This test gives us a broad overview of the blood, including the hemoglobin level, the number of red blood cells, and their size. In case of iron deficiency, the full blood count typically shows low hemoglobin and microcytic red blood cells, which are signs of anemia. The sample for this test is collected in an EDTA tube and is processed in a hematology lab. It is usually the first test ordered when patients present with symptoms like fatigue or weakness. The second test is reticulocyte count or retics. Reticulocytes are immature red blood cells, and this test helps us assess how the bone marrow is responding to anemia. In iron deficiency, the retics count is often low because the body doesn't have the necessary iron to produce new red cells efficiently. This test is collected in an EDTA tube and sent to the hematology lab at room temperature. Another vital test are components of iron studies. Iron, ferritin, and total iron binding capacity or TIBC. These tests are collected using an SST tube and processed in a biochemistry lab. In cases of iron deficiency, we typically see a decrease in iron and ferritin levels, indicating low circulating and stored iron. At the same time, The TIBC increases because the body produces more transferrin to capture whatever iron is available. Together, these results give a comprehensive picture of iron status and are crucial in diagnosing and managing iron deficiency anemia. Thank you.